In today's webinar, we're joined by Gift Nuka, who is going to talk to us about functional analysis of protein sequences using InterProScan. With that, I'm going to hand over to Gift and let him talk you through the webinar.
Okay, thank you very much, Melissa. My name is Gift Nuka. I work for the protein families team at the European Bioinformatics Institute, EMBL-EBI. And today I would like to present to you an overview of using InterProScan for the functional analysis of protein sequences as well as nucleotide sequences. And first I will look at the outline of my presentation. So I'll first introduce some of the main use cases for InterPro and InterProScan, and then I'll give a very brief overview of what InterPro is and what is InterProScan. Subsequently, I will look at the three ways in which you can use InterProScan, that is through the web interface or through the REST API. And finally, I will look at having a local installation that you can use on your computer to do sequence search. I'll also go through some of the outputs in the different output formats that we provide, and there are some differences, and you might be interested to know what those are. One of the use cases is, let's say you have isolated a new protein sequence, and you want to obtain a first-pass profile of the potential functions. So you have questions. What is the function of this protein? What family does? The others would be you have interest and you just want to know everything about it in terms of functional information. So what are the predicted domains for that particular protein? And what are the effects of those predicted domains or any other associated processes? Not a protein sequence, but you have a nucleotide sequence, and you want to all the functional information associated with that particular sequence. In this case, you might want to look at the protein families that would be given to that particular nucleotide sequence, or looking at the gene ontology terms that are associated with that particular sequence. In all these use cases, you would want to use the functional, and InterPro is one of those extensively used resource for protein function. So what InterPro does is it provides a functional analysis by classifying the proteins into families. It also predicts domains and important sites using information from different protein databases. Currently, we have 13 of these protein resources that we use in InterPro. And InterPro is heavily curated, so which means that information that we have has a literature references for it. What it does if protein sequence, so imagine that you have a linear sequence, and InterPro would be able to identify regions on the sequence and give them particular domains, or give them particular names to associate them as families, associate them as domains or important sites. So for example, if it can find a region that would be classified as a domain, InterPro would give an InterPro ID, which we call an InterPro entry for that particular domain, and it would give further information about the function of that particular domain. There are other InterPro entry types. For example, you could have active sites, which are a very fine-grained detail of the sequence, and you also have binding sites. You also have smaller units, which are repeated sequence units that are of importance. One thing that you can do, for example, to be able to search InterPro is to use InterProScan. And so behind InterPro, we have the software that you can use if you have a set of sequences that you would like to have functional annotations for. And what InterProScan does basically is that it looks each and every member database that is in InterPro, and it creates an analysis workflow. So for example, you have superfamily, you have PFAM, you have TigerFAM. For each database, it creates an analysis workflow. And at the end, when all the processing has been done, it creates a unified view of the functional annotations that it has been able to find. So for example, I can give a brief workflow for some of the member databases. So on the left, we have TMHMM, which is not exactly a member database, but it's one of the analysis features that we have in InterProScan. And it gives us predictions on some of the transmembrane features that you could have on a protein sequence. And on the right, we have for one of the databases, PFAM, and this is a much more complex analysis workflow. So if you look at the left-hand side, basically what you have is if you are given an input sequence in FASTA format, what we do in InterProScan is we run an algorithm against that sequence. In case of TMHMM, we use Hammer, and we just take the output that Hammer gives us and put that the unified view of the output. But with respect to PFAM, it's much more complicated because we have to do several post-processing steps. And in the case of PFAM as well, we have multiple PFAM modes, and in this case, it means that it will be much more involving. And after getting the output from Hammer, we do several post-processing steps, which basically would like to filter out those matches or those domains that are actually false positives. And in the end, of course, we combine all the outputs to create an InterProScan unified output. So this is an example of an analysis workflow that we have in InterProScan. So we have this workflow for each of the 13 member databases, also for the other feature analysis that we have in InterProScan. Okay, so I'll go on to look at what are the ways you can use InterProScan. There are three ways basically. One is to use the web sequence tool, and that's if you just have one sequence and you want to have the functional information for that particular sequence. The next way is using the InterProScan REST API. This also allows you to analyze one protein sequence of interest. And then the last way you can use is to basically install InterProScan on your computer, and you can run as many sequences as you can. So I will first quickly go through how you can use the web sequence tool. And basically, if you go to the website, let me see if actually I can. It will give you a box in which you can be able to cast and paste your sequence. And basically, if everything is working okay, let's just use sequence for analysis, and it will give you search. I already the protein sequence that I submitted, and summary information for all the InterPro functional information that I have for that particular sequence. So in this case, for example, it gives me the family, which I can hover off, and this is actually a hedgehog InterPro family, and also it gives me the segments that are predicted for this protein. There are many things you can do. So for example, if you want to have a copy of the output in TSV or the different formats that we have, you can go to the export button and actually get all that information. At the bottom, all the gene ontology terms that you can find. So in this case, we have biological processes, and it gives you the actual gene ontology, the names for those terms. So this is one way in which, and actually you can see this only allows you to search using one protein sequence, and it also doesn't allow you to use nucleotide sequences. So this is what I already presented. So you can paste your nucleotide sequence in the search box and get the results for that particular sequence. And these results remain on our servers for 7 days. So it means that if you have your protein search ID, you can use this and paste it in the import button, that is import search box that you can use, and then you can get these results again. But after 7 days, these results are deleted, and you would have to create a new search. Okay, I'll go on how you can use the InterProScan REST API. This allows you to download a script or create your own script that you can use to send sequences for analysis. If you go to the InterPro website and you go to InterPro scan, at the bottom you have the links on more information that you can use if you want to analyze sequences in this way. So for example, at the bottom here you have the link for the REST API documentation. If you go to that particular link, basically you will go to a page which gives you all the information that you need to know. So we have different ways in which you can use the web services. We have the REST. Of course, we also have, for those who are interested, we have the SOAP protocols, which nowadays are not so much popular. But with respect to InterProScan, we provide client scripts that you can use. So you don't need to write anything. You just need to download the client, for example, in Perl or in Python, and those scripts to send your sequences to our web servers. And you will get results for those searches. So if you know how to write REST API calls, basically we have a base URL. If you don't need to worry. You just need to download the script that I mentioned previously, using this particular command. So you just need to provide the sequence that you are analyzing. You need to provide the output file name you want to send all your results, and we also require that you specify an email. Basically, we want to be able to make sure that there's fair use of resources. So for the REST API, there's a limitation. You cannot send more than 30 sequences per time. So we want to be able to track those assertions. Okay, so here is an example of a run using the REST API. So I used the Python client, and I just downloaded the Python client from the website and provided my sequence and specified my output file name and also my email. And if you are running this interactively, basically what you have is it will the script will output the job name, so that if there are problems or if you need to get results, for example, if you want to get results from our website, you can just paste this job name into the button that I showed you earlier, and then it gives you the progress of how that job is running. And when it's finished, of course, it will create all the result files depending on what you had given as a name. And in this case, I didn't specify any particular limitations on the output, so it gives me everything that InterProScan can produce. So this is basically the way you can use the REST API, and it's much more straightforward. You don't need to download the complete InterProScan app. The script is actually very minimal. It's just a few hundred line code, so it will not occupy too much space on your computer. But using the web sequence search, using the REST API have their limitations. So while they have advantages, so for example, you don't need to download InterProScan. You are always accessing the latest version of InterProScan. You don't need to worry about maintaining a local copy. The fact that you can only use one sequence per time for the web sequence search might be a limitation for some users. Or in certain cases, for the REST API, even though it allows you to submit 30 sequences at a time, for certain use cases, that would not be scalable or not the optimal way to analyze sequences. And lastly, in both cases, you will not be able to analyze nucleotide sequences. So in that case, you might want to install the complete version of InterProScan, which is the command line tool that we provide. And our basically on how you can download InterProScan. This is a huge application. So if you look at the size of the release, it's about 9.2 gigabytes in a compressed format. In an uncompressed format, that would be around 40 gigabytes. So you need to have disk space for you to be able to run InterProScan. And actually, if you download the complete data that we provide, then you would probably need around double that particular amount of disk space. So I'll just go through how you can be able to download the application and run the application. So if you go to our website, as I said, basically all the links are provided. You can also use the instructions given on the documentation that we provide through Read the Docs, and it gives you all the steps you can go through to download InterProScan and actually run InterProScan. And it also has much more information about many other things that you would want to know about InterProScan. Okay, so here, basically, the most important part for you if you want to download InterProScan is just to either go to the FTP site and get the compressed form of InterProScan. And this is earlier on, so I have downloaded the application, and I also loaded the MD5 file for the application. Because it's a huge file, you just want to make sure that it is downloaded properly, and you can check that using the MD5 checksum. After downloading it, of course, you can decompress it using the tar command as mentioned here. And when everything is decompressed, you are ready to go. There's one other additional thing that you might want to download. We have Panther as one of our member databases, but it is a huge member database. So we provide Panther models separately, and so you have to download them in a different way. So this is the page from the Read the Docs that actually tells you how you can actually download Panther and be able to use it as part of InterProScan. I'll not go through downloading Panther here, but okay, so if you have downloaded InterProScan and you have everything running properly, you want to test if actually you can analyze your sequences. We provide test sequences included in the download. So there are two commands that we usually advise people to use. One is to just use the basic command, which is the first one. So you use the InterProScan command, and you specify the input sequence file, in this case, test all applications. We have this test all applications file because the output will hit all the analysis that we have in InterProScan. The second command basically has another option on it, which is disable pre-calculation. So in the first command, if InterProScan finds that the sequences you are using are already in the UniProt knowledge base, then it will not do any local computation for that. It will just get what we have already pre-computed, and that basically makes the analysis much more faster. In the second case, you want to test actually, because in most cases, people run sequences that are not in the UniProt knowledge base. So you would want to test if actually you can run and use InterProScan to calculate or characterize your proteins locally. And therefore, you have to use this option that says disable pre-calculation. So if I use the second command, it can look like this. It will tell you the progress that is being, if everyone successfully, it will say done. Okay, and that means that you can go and now look at the results. In this case, I look at what are the results that InterProScan generated from my particular run. The default output formats are 4 of them. So we have XML, we have TSV, we have JSON, and we have the GFF3. Okay, if you look at my commands, I apply the output format that I want. So in this case, that's why it just gives me all the output formats that are given by default. Of course, if you're only interested in one particular format, you can specify that. InterProScan has the option for you to specify the output format that you want. Okay, so as I mentioned, the default output formats are TSV, XML, JSON, and GFF3. But we also have other graphical outputs. These are HTML and SVG. There are some use cases where people want to have an HTML view or an SVG view of the output results. So for example, if you want to include the results in a paper, maybe you might want to use the SVG format because it provides a much more better way to do that than the other formats. Okay, so if the test sequence run was okay, then you can use your own sequences now to actually run InterProScan. And what I did was basically to try to look at run from our website, and we now have a proteome for SARS-CoV-2, which actually, if you go to our webpage, is identified on the homepage. And so I will try to run InterProScan against the proteome for SARS-CoV-2. And I got this proteome from our website by just following the links. So I go to SARS-CoV-2. It gives proteome page for SARS-CoV-2. And at the top here, you can identify, it says overview, entries, and proteins. And I'm interested in the proteins. So this proteome is a small proteome, so only 14 sequences in the reference proteome from UniProt. And the web page allows me to download that set of proteins using different formats, and I'm interested in the faster format. And what InterPro website does is it provides you with a script that you can actually use to download that particular data. And in this case, I use that script to download the proteome and run InterProScan against all the steps that I actually did. So get faster was my name of the script that I downloaded from InterPro, and I used Python 3 to get the proteome, and I named it SARS-CoV-2 proteome. I ran InterProScan, just the basic command, basically, provided the input and provided the output file that I want for this analysis. And after it finishes, I look at whether I have the result that I want. In this case, I also didn't specify what kind of formats I am interested in for the output, so it gives me all the 4 default formats that are provided. And here you can see I tried to look at many domains or families from this particular proteome, and I have 315 matches for this particular proteome. And just out of interest, I wanted to see how many of those matches are actually annotated with an InterPro entry, or so an InterPro domain, InterPro family, etc. And actually, we have 139 matches already annotated for this particular proteome. If I look at the that I get from InterProScan, I am interested in what are the matches, for example, that match one of the proteins in the SARS-CoV-2 proteome. This is the spike protein, which gives the SARS-CoV-2 its distinct feature. And I have several matches for that particular protein, and I have removed some of the information, but you can see in the first case, I have PFAM matches, superfamily matches, etc. Let me just look and zoom in on the first match that I have, and you can see that we have different functional information that we provide in the TSV format that can be useful. Apart from the coordinates that you can see that are in red, we also provide the Go terms, the InterPro entry names, the database signature, and the database itself for that particular match. We also provide other information. So, for example, we can provide the pathways that are associated with these domains. In this case, this particular match doesn't have any pathways, but I'll show that later on. And you can actually see that you have much more information than what can be provided by the TSV format. So if you look at the Go terms, it also gives you the particular strand of the Go term that is associated with that particular term. So you have biological processes, and you also have the actual Go term names that are associated with that particular match. So if you want more information, then maybe you would want to use the XML format or the JSON format, which gives similar information. One thing that InterProScan does is also it provides a more granular level of annotation. So it provides residue level annotations. And if you look at the XML format for one of the proteins, it will give me the binding sites. So, for example, it has the ATP binding sites, and it gives me the positions of those sites, which is useful for some people who want to have a more fine-grained view of the results. Okay, now I will look at different output formats, and in this case, I will use this the sequence that I had before, and if I run this sequence against InterProScan, it gives me more information than what I had with the SARS-CoV-2, because it also gives me the pathway information that are associated with the particular match. So as you can see, all the information in red is pathway information that would be useful. we also provide, as I mentioned before, HTML and SVG, but as you can see, the look and feel, if you have used our website, this look and feel is from the old website that is now obsolete. So I think what we'll do probably in the next few months is to be able to update the HTML and SVG formats so that they can reflect the new InterPro look and feel. But it also provides the same information as I mentioned before in a different way. The use case that I mentioned was using a nucleotide sequence, and basically we have a slightly more different workflow for this. What we have is that because InterProScan is primarily a protein search analysis tool, we have to do translation. So if you have a nucleotide sequence, we have to generate the open reading frames for that particular sequence, and then we use the analysis workflow that we have for the protein sequences. In the end, we have to map those results back to your original nucleotide sequence. One thing that you would have to do is to identify the type of the input as nucleotide, so -TN gives that information. By default, if you don't provide the type, InterProScan will assume you are using protein sequences. Okay, so if I run that nucleotide sequence, it will give me an output that actually I can use. And what I want to highlight here is basically the output has all the information that you would get in JSON, in XML, and it also gives you the cross references. So for example, in this case, you have the nucleotide sequence and it gives you the open reading frames relative to the nucleotide sequence. So you would want to know what position in the nucleotide sequence you have the open reading frames that you get. The next output for sequences that is useful is the GFF3 output. And here you can see basically, if you're familiar with GFF3, it provides with terms. So for example, in the first case, it gives us the idea that the nucleotide sequence had the length of 699, and the get-off was predicted on the complete nucleotide sequence. Then we have the rest of the matches that you can for that particular nucleotide sequence. I have removed some of the information, but basically this is the summary view that you can have for the GFF3 output. Okay, you have more options that you can use for InterProScan, and basically if you type help, InterProScan minus help, this would give you the options that you can use. And we have lots of documentation, either through read the docs for InterProScan or through our website. And if you have more information, if you want more information, basically you can go through to these websites. We have webinars, we have tutorials, and that would be useful for you to be able to use InterPro and InterProScan to analyze your sequences. Read the docs, as I mentioned, would be a useful resource for you. Okay, so I will provide the summary of what I've done. I've tried to give a brief overview of InterPro and InterProScan, and introduce the different ways that you can use InterProScan for analyzing your protein sequences. And I have presented some of the output formats that we have for InterProScan, and also that you can be able to use nucleotide sequences to analyze your sequences. Okay, now I would like to thank my team for the collaboration that we have, and for all the support they give for the work we do for InterPro and for InterProScan. And if there are any questions, I am happy to take those questions.
Thank you, Gift. There are, in fact, quite a lot of questions coming through the chat box already, so we can start on those straight away. So one of the first questions that came through is, what is meant by best score in clan filtering in PFAM search window? Workflow, rather, not window.
What is meant with clans and?
The best score in clan filtering.
Okay, so databases like PFAM organize their models in a way that if they find certain models have some similarities, they organize them into clans. And if we run InterProScan and we find that we have matches to models that actually come from the same clan, then we have to only choose one of the models as being the match for that particular sequence. And to do that, we use different scores. So we the actual score from Hammer that we use, or we can use the E values. Sometimes we also have to look at the length of the sequence region that is being hit. So we use those information to be able to filter the results that we get when we have matches that hit different models.
Okay, thank you. So the next question is, why don't some InterProScan outputs have an InterPro identifier?
So what happens is when we run InterProScan, we basically use the models that are from the member databases. And so it gives us information to say, okay, this particular model has identified this sequence to be a domain. And what InterPro does is that the curators for InterPro look at that information and try to see if actually it is correct or it makes sense. And if it does, then they create an InterPro entry for that particular signature hit. Sometimes curators might find that there are problems, and in that case, they will not be able to create an InterPro entry for those particular hits. So if you find that you have a match and you don't have an InterPro entry for that particular match, it either means the curators are still looking at it, or it has problems, or the curators have not just had the time, because basically some of the databases are very big, like Panther. It will not be possible to look at all the Panther models and integrate them into InterPro. So yeah, it's either of those three reasons when you don't have an InterPro entry on a match.
Okay, thanks. The next question is about the web tool. And the question is, can you only analyze sequences one by one using the web tool?
Yes, that's correct. Okay. So that's the limitation. So you can only analyze one sequence at a time. If you want to analyze more sequences, then you have to either use the REST API or actually download InterProScan locally.
Okay, so I think that probably partially answers the next question, which is, how can you scan a complete genome?
Yeah, so that's exactly the best way is to download InterProScan and analyze your genome locally, because that will provide you with all the flexibility of running against a genome.
Okay, and with the genomes, you were talking about having to translate it first. I guess that's something you'd have to do to analyze that genome first.
Yeah, exactly. So it means when the input is a genome, it will first translate first and then run through the protein sequence workflow and then eventually give you the results. But of course, if you use InterProScan and with XML and JSON, it will provide you with, or JFF, it will provide you with the mapping from the protein functional information that it has to the original genome sequences.
Okay, that makes sense. So next question is about the REST API, and the question is, can you run the REST API with 30 sequences in one FASTA file or with 30 separate files?
So yeah, that's the limitation for the REST API. You have to send one sequence at a time. So you cannot have 30 sequences in one file. So you have to have a way of being able to separate those sequences one at a time. Actually, there are tools that can actually help you doing that. So you don't need to actually separate them into physical files. But yeah, you have to do that. The reason why we do that is because of fair use. So we receive millions of sequence search job analysis, and therefore we don't have enough resources. So it's basically, we just want to make sure that each particular search is given the fair use it deserves.
Okay, that makes sense. The next question that I have coming through in the chat box is, one second, I just need to find it, is can we get the 3D structure of desired proteins from InterProScan?
Come again, sorry.
The question is, can we get the 3D structures of desired proteins from InterProScan?
So no, not from InterProScan. But if you go to the website, basically, you would be able to have 3D structures. So if you analyze one protein sequence, and as I gave you an example earlier on, you have a link to view the 3D structures of those proteins, but not through InterProScan.
So the 3D structures are hosted elsewhere?
So they are hosted at website, yeah. Because
sorry, where was that? Sorry, I didn't quite catch it.
Come again?
Where were they hosted? I didn't quite catch it.
So basically, it's the way the website works. So it gets this information, which is another resource at EBI, and so it has access to that data. InterProScan itself, if you run it locally, it doesn't have access to other resources that we have when you run or you search through the web tool. So that's the reason. Okay,
I see. Thank you. I can see there's been a couple of other questions about analysing things like mRNA and also something to do with RNA-seq, but I think you've already answered that in that for analysing nucleotide sequences, you first need to translate them into the protein sequence. Is there anything else that you wanted to add on that topic?
So basically, if you have a nucleotide sequence coming from any particular domain or in any particular biological context, we can analyze that. Yeah. You are the one who knows the context of that, and what we will do is we'll just be able to associate functional units, functional terms to what you provide us. We will not know exactly whether this is a DNA sequence, this is an mRNA sequence, whatever. We will just be able to take your sequence, translate it, generate the open reading frames, and then run the protein analysis workflow and give you the results. So yeah, it's possible as long as it's in the nucleotide sequence format.
Okay. So the next question is, can we do functional analysis of proteins on this platform?
Yes, of course, but I think that's very general, because InterProScan is basically a resource for functional analysis. But maybe the question would have to be more specific of what that means. I mean, InterPro is a functional analysis resource.
Perhaps the person who asked that question could put a bit more detail in the chat box if they want to know more, or you can get in touch with the InterProScan team as well.
If they don't have time now, they can write us an email and explain what exactly they want or mean by that.
Okay, are there any more questions this afternoon? Okay, so one more question coming in, and I just need a moment to read it. So the question is, are you aware of scripts to pass the InterProScan outputs? For example, to extract all domains overlapping a region?
No, we are not aware of any scripts that that do that. But we are welcome if people are interested in contributing. InterProScan is an open source application, so if people are interested in providing such kind of tools, then we would welcome that.
And there is also a question, can you use the API behind a proxy?
I think you can use the API behind the proxy. I haven't tested it. I would guess that you just have to provide the necessary details, because you can run InterProScan behind the proxy, and yeah, you just have to test it yourself. It's something that we haven't tested it. But I see no technical reasons of why it should not fail.
Okay, thank you. Any other questions? Just give it a moment or two. Okay, so there don't seem to be any more questions this afternoon. Thank you very much, Gift, for a great webinar. As Gift has mentioned, you can get in touch with the InterProScan team if you need more information or if you want to talk about your own work. This is also part of our regular webinar series, and you can find out more about upcoming webinars on the EBI and training website. So we'll leave it there for today. Thank you very much for joining us, and thanks again to Gift for presenting the webinar. Before we leave, sorry about that. Before we leave, just one quick little reminder to please fill in the feedback survey when you see it in your window. Thanks a lot and enjoy the rest of your day.